Let's establish something immediately. The image burned into popular culture, a 13-foot apex predator with a skull the size of a bathtub, is only half the story. The other half is sitting in your backyard right now, pecking at a piece of bread. What if I told you that the most rigorous genomic research of 2026 doesn't argue that birds descended from dinosaurs? It argues that birds are dinosaurs, not metaphorically, but in the strictest biological classification. The evidence is in their chromosome, and it's conclusive. When was the last time science found a 68-million-year-old fingerprint? In 2005, paleontologist Mary Schweitzer made a discovery that the field initially refused to accept: intact, soft tissue preserved inside the femur of a Tyrannosaurus rex. Not a mineral cat, not an impression. Actual flexible, translucent, biological material, and within it, extractable collagen protein fragments. To appreciate why this matters, consider what collagen is. It is a structural protein, the most abundant in vertebrate biology, and its amino acid sequence is extraordinarily conserved across evolutionary time. Unlike DNA, which degrades rapidly in open environments, collagen's tightly coiled triple helix structure resists decomposition under specific burial conditions. It is, functionally, a molecular time capsule. When Schweitzer's team ran a mass spectrometry analysis on the recovered T-Rex collagen and aligned those sequences against modern species, the results were unambiguous. The closest molecular match to a 68-million-year-old theropod dinosaur was not the Nile crocodile. It was not the Komodo dragon. It was Gallus gallus, the domestic chicken. This is not a loose statistical correlation. The collagen sequence homology between Tyrannosaurus rex and modern galliform birds is the kind of molecular alignment that, in any other context, would indicate recent common ancestry. In this context, it indicates direct lineage continuity. The smoking gun was found in a bone, and it pointed without ambiguity at every bird on this planet. Here's a question worth sitting with. Why do birds have approximately 80 chromosomes? The average mammal carries 46. Humans have 46. A domestic cat has 38, but the domestic chicken has 78. The zebra finch has 80, and this pattern, a high chromosome count with an unusual structural organization, is not random. According to landmark research published in 2026 examining avian karyotypic evolution, it is a direct inheritance from the theropod lineage. The key concept here is microchromosome. Birds possess a genomic architecture that is, to a significant degree, unique among living vertebrates. Roughly half of their chromosomes are classified as microchromosomes, small, gene-dense structures that carry a disproportionately high number of functional coding sequences relative to their size. These are not vestigial. They are extraordinarily active genomic real estate. The 2026 research traced the evolutionary origin of this microchromosomal architecture and concluded that the fundamental karyotypic structure observed in modern birds was already present in the earliest avian-adjacent theropods, approximately 240 million years ago, well before the Cretaceous-Paleogene extinction event that eliminated non-avian dinosaurs. Think of it this way. Imagine packing for an uncertain future. You could use a few large suitcases that are difficult to reorganize, or you could use dozens of small, highly efficient containers, each independently sealed, each capable of being rearranged without disrupting the others. Dinosaurs, specifically the theropod lineage, packed their genetic information in small, stable micro-suitcases. This karyotypic architecture proved to be extraordinarily resilient. When the KPG extinction event arrived 66 million years ago, asteroid impact, global firestorms, years of impact winter, the microchromosomal genome was structurally equipped to survive what larger, genomically inflexible lineages could not. The birds that exist today do not merely carry a resemblance to their theropod ancestors at the genomic level. They carry the same fundamental chromosomal blueprint, modified, refined, adapted, but structurally continuous. That is not ancestry. That is inheritance. What happens when you tell a bird embryo to stop being a bird? The answer, as it turns out, is deeply instructive. Over the past 2 decades, a discipline sometimes called reverse developmental biology has produced a series of findings that would be dismissed as science fiction if they were not meticulously peer-reviewed. The central figure in the popular science version of this story is paleontologist Jack Horner, whose Chickenosaurus project sought to demonstrate, through targeted gene suppression, that the developmental toolkit for constructing a dinosaurian body plan never actually left the avian genome. It was simply turned off. Here is what the experimental literature actually shows. Modern bird embryos, during a specific developmental window, initiate the formation of teeth. The genetic pathway for dentition, the same pathway present in toothed dinosaurs and early birds like Archaeopteryx, is activated in contemporary avian embryos before a separate molecular suppressor terminates it. The teeth do not form, but the instruction to form them does. Similarly, mutations affecting the TALPID2 gene in chicken embryos result in the development of small, conical teeth structurally consistent with those found in Mesozoic theropods. This is not a novel structure being generated from scratch. It is a deeply conserved genetic subroutine, one that has been present in the avian genome for over 100 million years, being expressed rather than suppressed. The same logic applies to tail development. Modern birds possess a pygostyle, a fused series of caudal vertebrae that anchors the tail feather. But when specific developmental genes are silenced in bird embryo, the result is a long, segmented reptilian tail. The genetic instruction set for that structure is present, active, functional. It requires deliberate molecular inhibition to prevent its expression in every bird that hatches. This is the critical insight. These are not theoretical connections between birds and dinosaurs. They are functional genomic continuity. Every fertilized bird egg contains, in its regulatory genome, the architectural plans for dentition, a reptilian tail, and forelimb structures more anatomically consistent with a velociraptor than with a modern wing. The dinosaur blueprint is not extinct. It is merely, in the parlance of developmental biology, transcriptionally silenced. Your backyard is full of dragons in standby mode. Here is where the story gets metabolically interesting. One of the persistent misconceptions about dinosaurs, perpetuated by decades of cold-blooded, slow-moving cinematic portrayals, is that they were physiologically similar to modern reptiles. The genomic and fossil evidence now tells a considerably different story, and birds are the primary exhibit. The genes encoding beta-carotene, the structural protein that forms feathers, are present in modern birds in a highly expanded gene family. This expansion is not observed in turtles, lizards, or crocodilians. It is, however, consistent with the fossilized impressions of feather-related structures found across multiple theropod species, from Anchiornis to Uanchuavi. The genomic architecture of the avian integument is a direct continuation of a lineage that was already experimenting with feathers, well before flight was a selective pressure. More physiologically significant, however, is the respiratory system. Birds possess a unidirectional airflow lung system, a network of air sacs that allows continuous gas exchange in both inhalation and exhalation. This system is, in terms of oxygen extraction efficiency, dramatically superior to the tidal breathing of mammal, and the skeletal evidence strongly suggests that derived theropod dinosaurs possessed air sac systems topologically identical to those of modern birds. Pneumatized vertebrae, hollow long bones, and interclavicular air sacs, inferable from preserved bone architecture. This is not incidental. The air sac system, when combined with evidence for elevated metabolic rates in large-bodied theropods, paints a picture of a physiologically dynamic animal, one that regulated body temperature endothermically, processed oxygen with exceptional efficiency, and sustained the kind of metabolic throughput that supports active, high-speed predation. Your parakeet, incidentally, has a resting metabolic rate roughly 6 times higher than a lizard of comparable mass. It maintains a core body temperature of approximately 41 degrees Celsius. It processes oxygen with an efficiency that no mammal can match. It also, if you have ever owned one, behaves with the absolute conviction that it is an apex predator. The genomics, to be fair, partially support this self-assessment. The KPG extinction event, 66 million years ago, eliminated approximately 75% of all species on Earth. Why did birds survive when Triceratops, Spinosaurus, and every non-avian dinosaur did not? The answer is not luck. It is genomic plasticity. The microchromosomal architecture discussed earlier is not merely stable, it is rearrangeable. Research into avian chromosome evolution demonstrates that bird lineages have undergone significantly more interchromosomal gene transfer and karyotypic reorganization than any comparable vertebrate group, while simultaneously maintaining the overall chromosomal structure inherited from their theropod ancestors. This combination of structural stability with localized flexibility represents an evolutionary advantage that is difficult to overstate. In practical terms, when the post-impact environment demanded rapid phenotypic adaptation, smaller body size, different dietary strategies, altered reproductive timing, the avian genome could accommodate those changes without catastrophic genomic instability. Large-bodied, genomically inflexible theropods had no equivalent mechanism. Their developmental programs were calibrated for an environment that no longer existed. The birds that survived were small, could exploit seed-based nutrition in a deforested world, and possessed a genome capable of rapid adaptive reorganization. They did not survive despite being dinosaurs. They survived because they were the most genomically sophisticated dinosaurs that had ever existed. Evolution did not start over after the asteroid. It promoted from within. So here is where we land. Every pigeon navigating your city is operating on a 240-million-year-old genomic platform. Every sparrow carries, in silenced regulatory sequences, the molecular instructions for teeth, claws, and a reptilian tail. Every egg contains a developmental program that predates the extinction of the non-avian dinosaurs by tens of millions of years. The asteroid did not end the age of dinosaurs. It curated it. The next time you see a pigeon on a ledge, that is a survivor of the most catastrophic extinction event in vertebrate history. Minimum respect is warranted.